We're going to discuss some aspects of digestive system in this lab today, and we're going to start with the general structure of the digestive system. So it consists of several parts: the mouth, pharynx, throat, esophagus, stomach, small and large intestine. And the function of the digestive system is to absorb, break down the food into the nutrients, and then absorb nutrients, vitamins, minerals, carbohydrates, proteins, and lipids from the gastrointestinal tract into the bloodstream. So here's the anatomy of gastrointestinal tract. It starts with the mouth, salivary glands and tongue assist in the initial digestion of food in the mouth. Then food gets to the esophagus, then in the stomach, and the small intestine. Then small intestine receives secretions from liver and pancreas, which you kind of can see here. Also, it receives bile squeezed out from the gallbladder, and then from small intestine it gets into the large intestine, and then feces leave your body through the anus. So we're going to discuss some gross anatomy in this exercise. We're going to start with the mouth, pharynx, and esophagus. So here's the mouth with tongue inside. Here's the oropharynx and laryngopharynx here, and that's esophagus. Stomach is right here. Small intestine, you can see here, that's the duodenum, that's jejunum. The darker colored and lighter colored is ileum. Three parts of the small intestine: duodenum, jejunum, and ileum. Then large intestine. So you've got cecum, ascending colon, transverse colon, descending, and sigmoid colon. And rectum that opens into the anus. So those are primary organs of digestive system, organs that form alimentary or gastrointestinal tract, while the accessory organs of digestive system include not only liver and gallbladder and pancreas, but also tongue and salivary glands, which are not shown on this picture. So which organ labeled A below? A is the small intestine. That's the place where nutrients are absorbed. Organ that is labeled B, responsible for the production of bile, is liver. Different sections are shown in these images. C, organ that stores the excess bile, right here, is gallbladder. And organ D is the stomach. You can get it from the hint that they give that this organ is connected to esophagus and the duodenum. Organ that labeled E is pancreas, because the head of pancreas resides within the loop of the duodenum. Now, digestion can be chemical and mechanical. In chemical digestion, special molecules called enzymes break down larger molecules into smaller molecules. And enzymes, as you can see, come out of this interaction unchanged. They basically assist the chemical reactions to run faster. In this example, large molecule is a substrate, and the small molecule is a product of the enzymatic reaction. Most enzymes are proteins. They have names like trypsin or ase. Okay, for instance, from the name you can figure out what enzyme does. Enzyme lactase breaks down lactose. Lactase breaks down lactose. The enzymes as proteins, they operate at the specific pH, specific acidity, and specific temperature. And if the conditions are not perfect, the enzymes may denature. they can lose their structure and therefore their function. So pH scale shown here, neutral pH is 7, pH of pure water. And then anything that is acidic is lower than 7, while everything that is basic is higher than 7. So we will try to, of course, there will not be any real experiments, but we will try to figure out what happens in some experiments in the following chapters. So there will be chemical indicators that will confirm that the reaction of digestion actually happens in this experiment. So first we're going to talk about lactase, the enzyme that breaks down lactose, the sugar found in milk. So when lactose is disaccharide. So when it is digested, this disaccharide is converted into two monosaccharides, glucose and galactose. Much like the little train that consists of two cars can be separated into each individual car. So the question is, how specific lactase is? Can it differentiate between three different disaccharides: sucrose, maltose, and lactose? Maltose consists of two glucose subunits, sucrose composed of glucose and fructose, while lactose, as we already saw, consists of glucose and galactose. So the hypothesis that we're going to suggest is that lactase, while being able to break down lactose, cannot digest sucrose or maltose. Now, here's the assembly. So we have 2 tubes of milk, 1 tube of sucrose solution, basically sugar, and 1 tube with a solution of maltose. Okay. And then we're going to determine how much glucose we have initially, baseline levels of glucose in each strip. Let's say it's 0 for the simplicity. Let's say it's 0, just to make it simple. And then we're going to add to each tube a certain solution. So we're going to add first tube distilled water. So basically our first tube here receives no enzyme. And these 3 tubes do receive lactase. Then we're going to mix the tubes and we're going to incubate them at 37 degrees Celsius for 5 minutes to let enzyme work. And then we're going to use glucose test strips. So we're not going to actually use specific numbers, but just make up. So if you mix milk and distilled water is not going to break down any milk. So we can easily say that it was initially 0, final concentration was 0. If we add sucrose, and we mix sucrose and lactase together, lactase is specific, so 0 and 0. And in experiment 4, it will be 0 and 0, while when we add lactase to milk sugar, the substrate that lactase is specific for, we're going to have, as a final concentration, let's say 300. Okay? So, of course, it's not a result, but you get the gist. So, lactase, can it digest variety of substrates? No, it can just one specific substrate. Lactase is the name of the enzyme that digests lactose. What's like the name for the enzyme that digests sucrose? Sucrase. And maltose, maltase. Again, A-S-E. And lactase deficiency is lactose intolerance. In treatment options, people just take enzyme supplements. Now, next quote-unquote experiment, we're going to talk about effects of pH on trypsin activity. Trypsin breaks down proteins. Breaks down proteins into small peptides, short chains of amino acids. And then pancreatic enzymes and other pancreatic enzymes and brush border enzymes can break down peptides into amino acids so they can be absorbed. Okay? So what we're going to try, we're going to use trypsin to digest milk proteins. White color in the milk is because of the casein, so that's milk prior to digestion, and then when we add trypsin, milk becomes clear. So here we're going to look at the role of pH in the activity of trypsin, and trypsin is found in the small intestine where pH is 8, so we're going to look at different pH. And we're going to use special reagent, all in quotation marks, that will be used to confirm that milk proteins were actually digested by trypsin. So here you can see that when there are no proteins or peptides, it is blue. When there are proteins, it is very dark blue, or peptides, it is red. So optimum pH for trypsin activity is 8. So our formulation is that trypsin will work best at the pH 8. So we have 5 tubes. We have one tube that is milk and water, or negative control. So does it contain proteins? Absolutely. So if it does contain proteins, we have dark blue. I'm going to put, not dark blue. Milk is white. Now when we mix milk and trypsin solution at pH 5, milk will be probably white with some traces. So will it work at pH 5? Probably so. Will they be proteins? Probably so. So it's going to be dark blue. And then at pH 7 and 8, most likely the milk will be clear. And the colors will be presence of peptides, red. So there will be peptides. What this experiment would show is that enzymes work best at the pH that is optimal for that particular enzyme. So for instance, at acidic pH, we're probably not going to have any result for presence of peptides, just the presence of proteins. Optimum pH for trypsin activity is 8. What would happen to trypsin if it was placed into acidic environment such as stomach? It would be denatured. And it would be denatured because of its structural arrangement, and that it exists best at the pH of 8. So many plant-based foods rich in starch, such as wheat, corn, peas, rice, potatoes, with an enzyme called amylase, which breaks down starch into di and trisaccharides. So when there is iodine binds to starch and gives it a pretty dark color. So when iodine binds to starch, it's pretty dark, but as starch gets digested, the solution with iodine being an indicator, it becomes more and more red. So our hypothesis will be that the optimal temperature for amylase is 37 degrees Celsius. And we have here a setup when we have just water. We have amylase at same concentration in the ice bath, the room temperature, the body temperature, and the high temperature. So after iodine addition, the water will be basically just brownish, yellowish, so just negative control. Now, the ice bath, amylase will not work. It will not be destroyed. It's just too cold. So the color will be probably somewhere between the brown, dark brown, and maroon. As we get to the room temperature, it will be more maroon to red. And the body temperature, it will be red. There will be complete digestion of starch. And at 80 degrees Celsius, there will be no digestion, so it will be purely brown because high temperature will inactivate amylase. The optimal temperature is 37. And yes, probably every enzyme in our body has the same optimal temperature because our body entirely at 37 degrees Celsius, except maybe for some specific enzymes that are closer to the skin. Now, effects of bile and lipase activity. So lipids, when they are digested, they have to be emulsified. And emulsification, since lipids are not water-soluble, when they are digested by lipase, they have to be emulsified by the bile to make them water-soluble. So here we have a reagent, okay, that shows you the pH of the solution. You can see that digestion of lipids produces acids. So complete digestion of lipids can lead to more acidic environment, making it yellow. And no digestion, basic pH. So biles will accelerate the lipid digestion. So here we have kind of an interesting mix. So we have a first tube, negative control tube, which contains cream, that's the source of fat, bile salt, okay, and phenol red. In the second, we have cream, so source of fat, no bile salt, and phenol red solution. And in the third one, we have same arrangements as the first, but in this case, in number 2 and 3, we add lipase. So negative control, cream, bile salt, phenol red. Initial color will be no breakdown, basic pH. Okay. So it will be, what's that? Red. We're going to do red, orange, and yellow. So red. Final color will also be red. Lipase without emulsifier. Will there be some breakdown? Yes, probably to the orange. And lipase with emulsifier go from red to yellow. So bile is necessary for lipid. Well, it is not 100% necessary, but it greatly accelerates lipid digestion. Can a person still produce bile if the gallbladder is removed? The answer is, of course, yes. The person cannot store or concentrate bile, but can produce it. So, okay. Isabel, abdominal pain, signs of indigestion, burning sensation in the belly. Physician did an endoscopy. So open sore in the lining of Isabel's stomach. This is peptic ulcer, ulceration of the stomach. Antibiotic therapy successfully treated disorder. The cause of Isabel's stomach sore is microorganism called Helicobacter pylori. Upper right portion of the abdomen, sharp pain means that in the area of the gallbladder, so hard mass inside of Maria's gallbladder, gallstones or cholecystitis. Common treatment, removal of gallbladder. Gallbladder is not a vital organ because it simply stores bile. People can live without gallbladder. So you see this mass. Now let's do review questions. So A's denotes enzyme. Teeth physically rip food into smaller pieces. This is an example of mechanical digestion. So okay, A is esophagus, B is liver, C is small intestine. Optimal temperature for digestive enzyme is 37. Organ produces by liver. That's gallbladder. Excess bile. Small intestine, pH of 8. That's basic. The substrate is proteins here. Enzyme is trypsin. Product is peptides. Pharynx. somewhere here. That's oropharynx, for instance. Salivary gland here. You can't really see any. Those are tonsils, so not salivary gland. Lactose intolerance is caused by deficiency of the lactase. Bile salts emulsify lipids into smaller droplets.